Well, good morning, everyone. Welcome. Thank you for joining us again. I'd like to recognize some special guests. We're joined by State Representative Kent Calfee and his wife Marilyn, somewhere in the audience. Here we go, down at the front. Start off by just saying a few words about the lecture series for those of you who maybe missed some of the earlier lectures. The Eugene P. Wigner Distinguished Lecture Series in Science, Technology, and Policy launched in November 2014. Actually, it launched on the exact 70th anniversary of the first criticality at the graphite reactor. So this has actually been the way that we're recognizing that milestone of 70 years since really, in certain sense, the inception of the lab. I mean, I guess we could have talked about when construction started and all that sort of thing, but, you know, if you think about it, since we're a research lab, we're all about doing science and engineering, and that really began here when the graphite reactor began operations. So I think it's an appropriate way to recognize it. Of course, it's turned out, I think, to be a very successful series if you look at the speakers that we've had to date, covering all three aspects of science, technology, and policy. So it may well be that we decide that it needs to be an ongoing thing rather than just a one-year recognition. And for that, I'm thankful to our corporate fellows who've played a key role in organizing this series. Of course, the goals are really to invigorate scientific discovery, spur technological innovation, and really participate in a productive science policy debate. And the lectures honor Eugene Wigner, who was kind of a patron saint of the lab. He trained as a chemical engineer, received the Nobel Prize in Physics in 1963 for atomic theory, made major contributions to our understanding of the role of symmetry in physical systems. He was a founder in the field of nuclear engineering and kind of a role model for the coupling of basic and applied work. It was in the period of time immediately after the Second World War, in 1946 and 97, that he played an important role as the research director of Oak Ridge National Lab, then Clinton Labs, and really laid a blueprint for many of the sorts of things that we continue to do to this day. Among Wigner's lasting legacies was the recruitment of Alexander Hollander to develop a biological research program at Oak Ridge. Like Alvin Weinberg, Hollander stuck around long after Wigner returned to Princeton, and at one time the biology division was by far the largest division of the laboratory. Biological research has remained a core capability for Oak Ridge National Lab, although it has evolved over time in response to changing scientific opportunities and also changing definition of the missions for the department as a whole. And our neutron facilities and the supercomputers have the capability to provide the biological community with an extraordinary set of tools. Now, of course, this is an argument that has been made in the past, and we have to be cautious about how far we go with that argument before actually demonstrating the value. But I think we already have a number of exciting results, both from our supercomputers, Titan, and previous to that, Jaguar, and some of the things we can do with the new instrumentation at both SNS and HIFER. And importantly, coupling those two capabilities together with a lot of the expertise that's resident in the biological sciences here at the lab in ways that help us solve important problems, whether they're in the DOE energy mission space or contributing to science more generally. So today's lecturer actually knows something about neutrons, although I don't think he'll be talking too much about that. He's a physicist by training and a biologist by inclination. I guess that's one way to characterize it. He has a Bachelor of Science degree in physics from Baroda University in India and a PhD in physics from The Ohio State University. He did graduate and postgraduate studies in biology at UC San Diego and in chemistry at Yale University. After a brief stint at Oak Ridge in the early 1980s, he moved on to Brookhaven National Laboratory's biology department, where he remained until 1995, when he became a professor in the biochemistry department at the University of Utah. Since 1999, he's been on the scientific staff of the MRC Laboratory of Molecular Biology in Cambridge. And in 2000, his laboratory determined the atomic structure of the 30S ribosomal subunit and its complexes with ligands and antibiotics. Portion of the research was actually conducted using the synchrotron, the APS, at the Argonne National Lab. For this work, Venki was awarded the 2009 Nobel Prize in Chemistry, sharing the award with Thomas Stites and Ada Yonath. The research has led to insights into how the ribosome reads the genetic code, as well as into various aspects of antibiotic function. In the last few years, Venki and his colleagues have determined the high-resolution structures of functional complexes of the entire ribosome at various stages along the translational pathway. In March 2014, they published a paper in Science describing their use of cryogenic electron microscopy to obtain the first ever glimpse of the structure of the mitochondrial ribosome at near atomic resolution. This paper has been described as one of the most exciting results in structural biology in recent years. In addition to the Nobel Prizes, received many awards and honors, Fellow of the Royal Society, elected in 2003, member of the National Academy of Sciences of the United States, elected in 2004, foreign member of the Indian National Science Academy, elected in 2008, and a fellow of Trinity College in Cambridge, elected in 2008. The lecture topic for today is how antibiotics block the ribosome, the cell's protein factory. Please welcome Venki Ramakrishnan.
Thank you. It's a great pleasure to be back here after so many years. I came here to give a talk in the mid-90s, but that's the last time I was in Oak Ridge, so that's almost 10, 15 years, no, 20 years ago. God, time flies. Anyway, I asked for this slide to be left on while we were getting started because I thought you could all look at it and figure out what various writers from different countries and musicians and a mathematician from Trinity College, actually, all had in common. And by now, I'm sure you've all figured out that they all died young, okay? And they all died young of infectious disease. And today, we have gotten into this mindset where we think that if we get a fever, we just go to a doctor, get a prescription, you know, it goes away and we go back to work or to school or whatever, and it's all fine. But, you know, those days are not actually over. There are really 100 million people who have active cases of tuberculosis today, and that leads to 2 million deaths a year. And you might think that TB is a problem of developing countries. Actually, that's not true. But in fact, it's becoming prevalent in the West as well because of its conjunction with HIV and AIDS. But there's another problem, which is Staph aureus, and that is a bacterium that likes to invade wounds after surgery. And it's called Staph aureus because aureus means gold, and you can see this gold-colored pigment. But when penicillin was discovered in the 1940s, essentially all Staph aureus would respond to penicillin, so there was no resistance at all. And very quickly in the hospitals, resistance started developing to penicillin. And later on, with a lag of a couple of decades, resistance spread widely throughout the community, so that today, nearly all Staph aureus is resistant to penicillin. Of course, there are other drugs that you can treat Staph aureus with, but, you know, the fact is that in Europe alone, 25,000 people a year die of drug-resistant Staph aureus, okay? And about a similar number in the U.S., because they're roughly the same size. And so this is, you know, from the Financial Times. And you may recall that there was an article in The Economist only about 2 months ago saying that the drugs are not working. And now, it's not just Staph aureus, urinary tract infections, sexually transmitted diseases. All of these things are now becoming resistant to all possible antibiotics. And a couple of years ago, there were cases of what's called TDR TB, which is totally drug-resistant TB. There's no antibiotic that TB responds to. So you can see that it's a serious problem. And this leads to the question of what these antibiotics are and how they work and what we could possibly do about this impending crisis. And there isn't a single solution, but I'll give you some idea, and especially the kinds of antibiotics that target the molecule I work with. So people have known for a long time, probably for centuries, that if you took certain things, you would feel better. And in hindsight, there were a kind of antibiotic. But the modern idea of targeting chemicals to bacteria, you could say started with Paul Ehrlich, who was a very famous German microbiologist. And the idea occurred to him because he noticed that when he stained bacteria, he took infected samples and looked at them, he realized that certain stains would stain these rod-like bacteria preferentially. And so they're dark red because they've absorbed the red stain. And these large human cells are relatively unstained. And so he realized that there were compounds that would preferentially bind to bacteria. So then he took the next step. What if these compounds were actually toxic? So then those compounds would preferentially bind to bacteria, and they would kill the bacteria. And they wouldn't bind to the human cells, or at least not as well, and so the human cells wouldn't die. And so he called this the magic bullet. It's a bullet that only attacks your target. It only kills the specific target. Now, he himself wasn't so successful at producing a useful compound. He produced an arsenic compound that had lots of toxic side effects. But somebody who was influenced by Paul Ehrlich was another German, Gerhard Domack. He worked for the Bayer Chemical Company, which is famous for making aspirin. Okay, it was the first company to make aspirin. But it also, because it was a chemical company, had made lots of, thousands literally of compounds, many of which were dyes, organic dyes. And he systematically screened, in a very Germanic way, he systematically screened hundreds or thousands of compounds for their antibacterial properties. And he came up with Prontosil, which was a red dye. And this drug turned out to be the first highly effective drug against lots of fatal diseases. So in those days, if you got a cut, or even if you were working in the garden, in the soil, and you got infected, the infection, streptococcus could spread through your limbs, and there was actually no cure. You'd have to amputate, your limb if you got really infected and you couldn't fight it off. And what happened to him was interesting. His own daughter got her finger infected soon after he discovered Prontosil, and he saved his own daughter by treating her with Prontosil. He persuaded the surgeons to let him treat her with Prontosil before they decided to amputate, and she recovered. But he was unlucky in a number of ways. One of which was that a year after he had discovered Prontosil and patented it, it was found that the active ingredient wasn't Prontosil at all, but rather when you took Prontosil, it was broken down in your body into two compounds, one of which was the active compound, which was a colorless compound called sulfanilamide. Now, you may have heard of sulfa drugs. Well, sulfanilamide is the first of the sulfa drugs, okay? And it was, sulfa drugs are still used today, for infections which are resistant to, penicillin-based or other kinds of antibiotics. Anyway, so that was the first bad luck. The second bad luck was that he was awarded the Nobel Prize in 1939. If it had been awarded it 10 years earlier or, 10 years later, it would have been fine. But in 1939, Hitler had banned Germans from accepting the Nobel Prize because a few years earlier, the Nobel Peace Prize, which, as you know, is given in Oslo and not in Sweden, was given to a German pacifist who was against the Nazis. So he forbade Germans from accepting the Nobel Prize. Presumably he didn't know the difference between science and peace or between Sweden and Norway. But anyway, that was the rule. And so, in fact, so much so that, the Gestapo briefly arrested Domak, before they let him go. Because they thought, he was anti-patriotic, because he got the Nobel Prize. Anyway, after the war, the Nobel Foundation invited him to, their next Nobel Prize ceremony, which had been suspended during the war. And they handed him his check and his medal, and they said, we're very sorry, but because so many years have passed, the prize money was returned to the general funds, and there's no mechanism to re-extract it to give you the money. So there he was. But anyway, he didn't have patent money either. So, however, he had the satisfaction of being the sort of father of the modern antibiotic movement and having saved millions of lives, which I think is probably worth a lot more. Now, Domak worked by screening synthetic compounds, but another strain of antibiotic discovery was discovered by accident by this Scottish microbiologist named Alexander Fleming, whose mother told him that, look, if you stick around in Glasgow, you'll have to compete with other Scots. But if you go off to London, you'll only have to compete with the English, so you're better off going to London. And so and this is an issue now because, Scotland's going to decide whether to secede from the UK or not this September. Anyway, he went off to London, and one day he found his plate was infected by a mold spore. And this mold spore, it got contaminated. And he noticed something interesting, and that is that these bacterial colonies grew very well if they were far away from the spore, but as they got closer, the colonies kept getting smaller, as if the spore was somehow secreting something that was inhibiting the growth of these bacteria. And he correctly surmised that the spore, which was penicillin, which is like a kind of a bread mold, was secreting something that was killing bacteria, or at least stopping them from growing, and he called that compound penicillin. And it took a lot of time. This was in 1929. This is the original plate, actually. But it took until about 1940s, early 40s, to actually make penicillin in sufficient amounts to be able to treat people, because it was a huge effort, and it was only brought on because more people were dying of infection in World War II than from bullets. Okay, and so that was an effort that was initiated at Oxford by Chain and Florey, who went on to share the Nobel Prize with Alexander Fleming. And the discovery of penicillin from a natural compound, natural product, so his mold was simply making this stuff, led Selman Waxman, who was a professor at Rutgers. He was a soil microbiologist, and he said, well, if this guy can get stuff out of mold, I know how many types of bacteria there are in the soil, and so maybe soil bacteria are making things too. And he persuaded his student Albert Schatz to systematically survey a bunch of soil bacteria. And Schatz, after several years of painstaking work, identified Streptomyces griseus, a soil bacterium, which produced something that seemed to inhibit a wide variety of bacteria. So the thing with penicillin was it would only work against a certain class of bacteria called gram-positive bacteria. The gram-negative bacteria wouldn't take up the penicillin, and their cell walls were different. So, so it wasn't universally effective. But this compound was effective against everything they looked at. And so they asked whether it would even work against TB, which has a very complicated cell wall and cell membrane. And they found that, in fact, it could actually inhibit TB. So this was the first effective modern antibiotic against TB. It's not used much anymore because it has problems. TB very quickly acquires resistance to it, and it does have some side effects. But it was the first effective drug for which Selman Waxman received the Nobel Prize. And the problem was he didn't share it, even though the patent was with Schatz and Waxman. And so the result was that Schatz actually sued Waxman. So this is a thing that you shouldn't, you should be nice to your graduate students, otherwise one day they might sue you, okay? And, history has vindicated Schatz's view, actually. I think most people today believe that really they should have shared it. Anyway, the fact that soil bacteria could produce a compound like streptomycin, which would be effective, led to a systematic search largely from soil bacteria, and in fact, a lot of it came from a single genus, Streptomyces. And that resulted in a huge number of antibiotics. Many of these were discovered then by pharmaceutical companies, which felt, this could be a great way to discover new antibiotics. And a lot of them were discovered by Pfizer. And so what these antibiotics have in common is that they all work by blocking protein production in bacteria. So this, of course, leads to the question of how proteins are made, by bacteria. So I'm going to now switch to molecular biology, which means I'm going to start showing you molecules. And I want to show you, I want to get into your head, those of you who are not chemists or biologists, that we can show molecules in different ways. We can show them in a very realistic way, or we can show them in what I would call an iconic representation. And so I'm going to show you a bipartisan view of how we show people, okay? So here are a couple of people, and what we're seeing is a photograph, which is a very realistic way of showing them. But we could also show them in a simplified form, okay, which is like a cartoon, but they're still recognizable as the people. So you can see features that are characteristic of the person. And if you looked at those faces of those cartoons, you'd immediately know who you're talking about. But sometimes you can go simplify even further, and what you capture is some essential characteristic or feature of the person. And so here are some essential characteristics. One person is a waffle because they can't make up their mind. And the other person is all hat and no cattle, okay? So that's, some essential feature that if you know the context, you can figure out what they mean. So it's just the same way with proteins, okay? So you can show them as atoms, which are what proteins are made of, or you can show them as these schematics, which just tell you a little bit about the architecture or the topology of the molecule. But of course, you have to know that all of the atoms are still there, even though you're showing a simplified representation. And so these are three types of proteins. This is a protein that makes up our skin and connective tissue. It's probably the single most abundant protein in our bodies, and it's called collagen. Without it, we would just be a lump of jelly and just a blob. We would have no structure. Now, this is a protein that carries oxygen in our blood. It takes the oxygen, it captures the oxygen in our lungs and carries it through our blood into the muscles. And it's one of the first proteins whose structure was solved by the person who founded my institute in Cambridge, for which he received the Nobel Prize in 1962. And this is a protein that senses light, and it's present in all our eyes, and without it, you wouldn't be able to sense light. okay? So it's just to show you that proteins do hundreds of things, okay? Here are 3 completely different things that proteins do. And the question is, these antibiotics stop bacteria from making proteins. Proteins do hundreds of things, and they're essential for life. And so how are proteins made? Because we need to understand that before we can see how they're stopped from being made. Now, this is a rather old figure. This is taken from 2001. This is the genome. When we talk about the genome, what we mean, people know about the human genome or the bacterial genome. What they really mean is the entire sequence of information that consists of all of the genes in the organism. And the human genome consists of, it used to be thought 35,000 because we thought we couldn't be that much. We couldn't, if a worm has 20,000, but then we should have a lot more genes is what the thinking was. But actually we don't, so we have about 20,000 genes just like the worm. Now if you look at what the gene looks like, it's essentially in the form of DNA, which in that cartoon form is a double helix, 2 strands of a double helix with 4 types of building blocks. And the important thing about these building blocks is each building block only will pair up with a particular partner. So there are 4 types of building blocks called A, T, C, and G. But if you look at them in detail, because of the bonding pattern, T will only bond with an A and G will only bond with a C, okay? And the result is that if you know the sequence of building blocks on one strand, you'll automatically know what the sequence is on the other strand, okay? So this is how these 2 strands are complementary. And it also means that when these strands split apart, you can use the information in each strand to make the complementary strand, and that's how life essentially can carry on and divide. Now, this sequence is what specifies a particular protein. And the way it works is that each strand on the DNA has a sequence of these bases, which are those 4 types of building blocks that I showed you. And that gets copied into a molecule called RNA, and that then, gets converted into a protein. But the point is that RNA and DNA are very similar in that they're both what are called nucleic acids with 4 types of building blocks. And they're nucleic acids because they have a sugar and a phosphate group alternating in terms of this chain, and then these bases are attached to them. These 4 types of bases are attached to them. But proteins are very different. They're a completely different kind of polymer, and there are 20 types of amino acids that typically make up a protein. There are actually a couple more, but the other 2 are only used in special cases. So how do you go from the information in the gene to the information to actually making a protein that this gene is coding for in some way? So the way that works is one of the strands, the strand that contains the information in DNA, is copied. Now, there are only 4 types of these building blocks. So if each of them specified an amino acid, you'd only be able to use up 4 amino acids, okay? And if you read them 2 at a time, then you could have 4 times 4, or 16 possible combinations, okay? That's still not enough because there are 20 types. So you need to read them at least 3 at a time. And interestingly, this was figured out without knowing anything about it by a physicist named George Gamow, okay, who was interested in the coding problem and hung out with all these molecular biologists like Watson and Crick. So the way it works is that each group of 3 bases is a unit of information called a codon, and each codon then specifies a particular amino acid. And what Crick figured out, or actually theorized, was that there would be an adapter molecule which would somehow recognize the 3 bases, perhaps by base pairing with them, just like the 2 strands on the DNA are base paired. So these bases would again be complementary. And so each base on this adapter molecule would pair up with a particular complement on the codon on the gene, and it would bring along its particular amino acid. And then the next codon would have its own adapter, which would bring another amino acid, and somehow these things would get stitched together to make a protein. Now, of course, somehow isn't quite so simple, because it doesn't just happen by itself. It was found out by cell biologists when they were looking at proteins, where proteins were made. And they did this by giving radioactive amino acids to cells and then stopping them suddenly and then asking, where did labeled proteins end up? And they found out that these labeled proteins ended up in these particles on the endoplasmic reticulum. And when the particles were isolated, they turned out to be roughly the same in size. So it's not like there were all different types of particles, but they all looked as though they were the same. And they were about 250 angstroms in diameter. And as you can see, they all consisted of a large and a small half. So if you look at each of them, they're bipartite in structure. And because they were from the microsome and they consisted of RNA and protein, they were about 2/3 RNA. They were called the ribonucleoprotein particles of the microsomal fraction. Now, this is a mouthful to say every time you want to refer to it. So in the late 50s, somebody said, why don't we just call it the ribosome? And that's the name that's stuck ever since. And if you look at the ribosome, it was analyzed and shown to be made up of more than 50 different proteins, which assembled into 2 subunits, which then formed the entire ribosome. This is from bacteria, and the ribosomes in humans or even in yeast are much larger. They're almost 50% larger than the ones in bacteria. So how would the ribosome work? Well, you have the gene, and the DNA is copied into one of the strands is copied into this kind of temporary piece of information called messenger RNA, which you can think of DNA as the archival material. If you go to the Library of Congress, they're not going to let you take the book out, okay? But they will let you, they will give you a photograph of the book, like a Xerox copy of the book that you can look at. So you can think of RNA as a working copy, and the DNA as the archival material. So, but the genetic information is there, just as it is in DNA. And so what the ribosome has to do is figure out where to start, and that requires some special accessory factors, and it requires a special adapter molecule. These adapter molecules are called tRNAs, which recognizes the first codon. This is where the protein information begins, okay? And then it has these slots for the tRNA molecules. And so the next tRNA molecule, which is the right one corresponding to this codon, brings in the appropriate amino acid. And then the ribosome stitches a bond between those 2 amino acids and then moves. And after both halves have moved relative to the message, first tRNA is discarded, and now the new codon has been brought into this slot, so the next tRNA can come in, bringing in the next amino acid, and then you can have a bond between the second and third amino acid, and so on. So you can see what's happening. It's reading the information here on the gene, and it's stitching together the protein in a completely different part of the molecule. So it's like a complicated ticker tape reader, okay? So you've got this ticker tape that's threaded through one part of the machine. It's reading the instructions in the ticker tape and making what it's supposed to make, which is this polymer in the other part of the machine, in a completely different part of the machine, okay? Now, that's a very schematic, cartoony version, okay, of what the ribosome looks like. And this is what the ribosome looked like by electron microscopy about mid-90s, so about 20 years ago. And you can see already that it's much more complicated. So the RNA actually snakes around a cleft in the small subunit. And this is where the codons are presented to the tRNAs, which recognize them. And this is the other end of the tRNAs, where the amino acids are brought in. And this is the tRNA that's holding the growing protein chain. And this is the one that was bringing in the new amino acid. And so now this amino acid has to be added to the growing protein chain. And that's done by transferring this entire protein chain to the new amino acid. And then the old amino acid's kicked out and the new amino acid moves in to its place. And, the whole thing just keeps on going. But the point is that if you want to look at this in more detail and you want to understand how antibiotics bind to it, you need something like an atomic structure of this machine. So it's a little bit like, if you want to understand, if you were a Martian and you looked at the Earth and you saw cars moving around, you would know that they consumed gasoline and generated heat and carbon dioxide and water and some pollutants. But you wouldn't know how they really worked unless you knew in detail what a car looked like. And to do that, you would have to open up the hood and you would have to see all of the moving parts. And then that would only give you an idea that you would actually have to see it in movement. Okay? So we would want to look at the ribosome in great detail. And so that leads to a question of how you look at objects in great detail. And the classic way to do that is what we would call microscopy, which is you would take an object and put a lens in front of it. Now, if you think about what a lens does is take scattered rays from the object and recombine them to an image. And that image can be a magnified image, or it could be a much smaller image. So if you use a telescope, you're looking at an image that's a lot smaller than the object. Okay? But if you look at a magnifying glass or a microscope, your image is much larger than the object. Okay? So it all depends on how you set up the lens. But the point is that the scattered rays are there whether the lens is there or not. Okay? All the lens is doing, so the information is, if you take away the lens, then the information is here whether the lens is there or not to reconstruct the image. All the lens is doing is recombining the scattered rays to construct the image. So for those of you who understand it, the lens is effectively an analog Fourier transform device. Okay? So, and right now, all of you are doing exactly this. You're sitting here, you're looking at the screen, or possibly looking at me. You should be looking at the screen, actually. So, and the scattered rays from the screen are being recombined by the lens in your eye to form an image in your retina. But I notice there are some empty seats. And the point is, the scattered rays are going to those empty seats as well. And if somebody were to come and sit there, they would also be able to see an image of the screen. So that's the important thing to keep in mind. Now, the problem with microscopy is that if you use light, then light has a wavelength of 500 nanometers. And the distance between atoms is about 0.2 nanometers. And so you cannot, even with modern optical techniques which are trying to defeat this limit, you cannot actually see details which are much different from half the wavelength of the light. It's a bit like saying, you want to measure, some unit. If you were to take a big yardstick with no small markings, you wouldn't be able to measure anything that was much smaller than the yardstick. And so it's somewhat analogous to that, okay? But it really comes from a law of diffraction, that you can't see objects that are closer together than the wavelength of half the wavelength of the light that you're using. Now, there is light of the kind of wavelength that's appropriate for looking at distance between atoms, and those are what we call X-rays. Okay, so X-rays are just light rays, but with very short wavelength. Now, there are two problems with X-ray microscopy. Firstly, X-ray microscopes don't have, they're not as well built, they don't have the kind of properties that light microscopes have. We do have X-ray microscopes, but they work on soft X-rays. You don't have very good microscopes for hard X-rays. The other problem is that a much more serious one, and that is, unlike light, which doesn't light hitting the screen doesn't damage the screen. But X-rays actually do damage the object that they're looking at. They cause electrons to leave and they ionize the object. And so this is a problem. This is why you're not supposed to be exposed to lots of X-rays. And so if you were to try and get enough signal from a single molecule, you'd have to hit it with so many X-rays that you'd essentially destroy the molecule before you can see it. And people are trying to find a way around it with what's called the free electron laser, where you collect all your data before you destroy the molecule. But that's still a long way to go before it's applicable to single molecules as opposed to crystals. But in the meantime, for the last 100 years, we've been able to use X-rays in a different way, and that is by crystallography. This is a technique that was invented and also developed over many years, over 50 years, in the place where I work, which is in Cambridge. And in that technique, you form regular three-dimensional arrays of your molecule, kind of like stacking, oranges into a very symmetric regular array. But you're doing this with molecules. And you then shine your beam of X-rays onto these crystals. And what happens is, because you have a regular array, all the molecules interfere with each other. The scattered rays from each of the molecules all interfere, and that results in the scattered rays only showing up in certain directions where the interference is constructive. And they show up as spots. So they don't show up everywhere like these light rays from the screen, but they show up only along certain directions, which are called reflections, because they were originally formulated as reflections from the planes in a crystal. Now, the point is you can measure these scattered rays very accurately, and then you can do in a computer what a lens does in real life, which is you can recombine the rays to form an image of the object. Now, for this, you need very powerful sources of X-rays, and especially for the molecule I work with, which is a very weakly diffracting object, you need extremely powerful sources. And this could only be done with these third-generation synchrotron sources, which produce very powerful beams of X-rays. And these are some of the synchrotrons that my lab used, and of course there are others throughout the world now. And this is what the inside of a synchrotron looks like. So this is a huge building, a huge ring, and you can see, I don't know if you can see cars. Here are some cars parked here. So that just gives you an idea of the scale. And this is actually a big multi-story building. And so there are actually dozens of instruments around the ring, each of which is set up to do a particular experiment. And if you look at it, here's the inside of one of those instruments. They're called hutches. And of course, you don't want to be in there when the experiment's being done because of the dose of X-rays. And the X-rays come out through a pipe here, and they hit the crystal, which is being kept cold by a stream of nitrogen at very close to liquid nitrogen temperature. That prevents the crystal from being damaged very rapidly. It slows down the damage. And then the scattered rays are detected by a detector. We have better detectors now, but when we solved the ribosome, we were using CCD detectors. And it's an interesting coincidence that the people who developed CCD detectors got the Physics Nobel Prize the same year that the ribosome got the Chemistry Nobel Prize. And so the chemists couldn't possibly have gotten their prize if it hadn't been for the physicists having developed their CCD detectors at that point. So, well, it might have been, but it would have been a lot tougher. So anyway, when you do the experiment, what you get is an image of the object. Okay, just as with microscopy, you get an image of the object. Now, this image doesn't tell you what the structure of the molecule is. It doesn't tell you where the atoms are. It's simply an image, okay? And you have to figure out from looking at this image what the structure of the molecule is. And this is a little bit like solving a large jigsaw puzzle, okay? But there are some interesting differences. The first is this is a real experiment. So this means that there are what we call error. The image is not perfect. There are errors in the image. It means that because of noise, what we call noise, parts of the image actually don't belong in the image, okay? And we have to know that they're not really part of the image. They're just experimental error. And then parts of the image are missing. This could be due to the experimental error, or it could be because the molecule itself is moving around from one molecule to the next in the crystal, and so the image is blurred out. So this would be like buying a jigsaw puzzle and finding out that, there are pieces there that don't belong in that puzzle. They're from some other puzzle. And there are also pieces of your own puzzle that are actually missing. Okay, so it's a little bit like that. Now, the other difference, of course, is that this is a three-dimensional puzzle. It's not a two-dimensional puzzle like a jigsaw puzzle. And the third difference is perhaps the most important one. When you go buy a jigsaw puzzle, they very conveniently give you the answer on the cover of the box. Okay, and we don't know what this molecule looks like. So that's a problem. But you solve it in the same way, in a similar way, and that is, if you've solved a jigsaw puzzle, and my wife is an avid puzzler, what you do is you collect all the edges because you can recognize them, and you solve the edges first. And then you look for areas which have a very recognizable pattern, and then you build those. Then you save, sky and grass and stuff like that for the very end, okay? And that's exactly what we do. We would zoom in, and if you zoom in, suddenly what you see are like these two ridges, okay? So you have these two ridges, and you can see very regular bumps, okay? And there's bumps on the other side too. And somehow these ridges seem to be connected to each other. And if you know what you're doing, what you realize is that this is a double helical piece of RNA. Okay, so these are the sugars, these bumps are the phosphate groups, and then these connecting parts are the base pairs. Okay, so suddenly you've taken a piece of the image and you've figured out what it's like in molecular terms. So you've interpreted it as a molecular structure. And now you have to do that for the whole image, okay? So first you have to zoom out and figure out which part it is that you recognized, and then you have to build the whole rest of the molecule, okay? So, and that's it essentially takes, for large objects like this, it can take weeks or months of painstaking work in front of a computer. And I've had people who spent their entire lives in darkness for, several months, okay, because they're in this dark room with this stereo graphics terminal. So doing exactly this, both the large and small pieces of the ribosome were solved in 2000. And one of the puzzles about the ribosome was it's 2/3 RNA, but it has about 50 different proteins. And this is a protein-making machine. So if you have a protein-making machine that itself is made of proteins, where did the ribosome even come from? Okay, how could a protein-making machine itself be made of proteins? Well, the answer to this is that Crick thought that maybe early ribosomes consisted only of RNA, and then, proteins came later. And many biologists believe life began as an RNA world that consisted mostly of RNA, because RNA is the only molecule that can carry both information and therefore can replicate, and also do catalysis, okay, like protein enzymes. And whereas proteins can't carry replicating information, they can in some special cases like prion diseases and so on, but not in a general way. But they can do catalysis. And DNA can carry information, but it can't do catalysis, okay? But RNA can do both. And the interesting thing from these structures is that the important parts of the ribosome were part of a very highly conserved core that consisted entirely of RNA. By conserved, I mean this core is the same in humans and in bacteria and, in worms and everything, okay? It's very, very similar. Okay? So the ribosome suggested that maybe it started off as just RNA and then became this. So then a few years later, we solved the entire ribosome. This is about 500,000 atoms, both pieces of the ribosome with the tRNAs in between. Once you've solved the ribosome, it's very easy. Solving the ribosome was technically hard, But after having solved it, it's easy to solve how antibiotics bind to it. And so you can do the same experiment and get the image with the antibiotic and then subtract away the rest of the ribosome. And what you're left with is the image of the antibiotic itself. And so here's an antibiotic called spectrinomycin. It binds in this tiny little pocket here in the ribosome. And because this head is supposed to move during the ribosome function, it kind of locks the movement of the head and doesn't allow the ribosome to to move, and so it stops the ribosome dead. And using this method, we solved the structure of a large number of antibiotics. And while we were doing that on the small subunit, Tom Stites and Peter Moore and their colleagues at Yale was looking at the structure of the large subunit, and they solved the structure of a number of antibiotics from the large subunit. Now, large subunit is where the protein is made, where the amino acids are stitched together to form the protein chain. And that happens in the peptidyltransferase center, and an antibiotic called chloramphenicol binds there. And then there are other antibiotics that bind at the mouth of this tunnel through which the protein chain has to come out, before it's released from the ribosome. So all the proteins that are made have to come out of this tunnel, okay? So when you look at the ribosome, it looks like this. Here's the large subunit, small subunit. These are the tRNA molecules. But if you now zoom in to the peptidyltransferase center where the amino acids are linked together to form the growing protein chain, here's the first tRNA, and here's the new tRNA, which brings in the new amino acid. And the ribosome has to start by making a bond between these two amino acids. And chloramphenicol binds right here. So it would prevent the new amino acid from even being able to enter the site where it can then be added on to the first amino acid. So it stops the bonding, the stitching together of the protein chain, and that's how chloramphenicol works. And erythromycin and azithromycin and other whole class of compounds called macrolides bind at the mouth to the tunnel, and presumably they work by stopping the protein chain from being able to go through the tunnel, by kind of constricting the tunnel and not allowing the protein chain to go through. Now, these two antibiotics have problems. So this is just to show you what they look like. Now, the discovery of the ribosome structure and these antibiotic structures led Stites and his colleagues to form a company in New Haven called Ribex Pharmaceuticals. And they decided to use these structures to try and design new antibiotics. And one of the things they noticed was that the chloramphenicol site and the azithromycin site were right next to each other. And there are problems with both antibiotics. Chloramphenicol is no longer used. I'm probably the only person in this room who's had it because it's highly toxic. It used to be given in India if you had typhoid because it was effective against typhoid. But it has serious problems. It causes anemia. It can cause ototoxicity, so you might need hearing aids prematurely and so on. So it's not widely in use. But the other problem, azithromycin and erythromycin are pretty well tolerated and widely used antibiotics, even today. But the problem is lots of bacteria have become resistant to macrolides, like erythromycin. So these guys at Ribex had the idea, how about if we make a molecule that spans both of these sites so that it binds to both sites? Now, such a molecule might not have the side effects of chloramphenicol, which is a small molecule that could bind elsewhere, or it might not have the resistance. It might be able to overcome the resistance of erythromycin. So they made this molecule, which they called RX-2102, and they found it binds exactly the way it did, the way they thought it would. And this is a molecule that now is effective against all of the resistant strains that they had previously tested. So the story would be nicer if it weren't for the fact that the current business model for how to make antibiotics is broken. Okay? So this company tried to launch an IPO a couple of years ago, and the share price that they could get in an IPO was much, much less, a tiny, small fraction of what the investors had already put into the company. Okay, so the IPO was withdrawn, then it got bought by another company, and they're still trying to make a go of it. The very same month, a company that is used to share gossip and photographs of your, girlfriend or your kid or something was worth billions. Okay, so there's something wrong with a situation where a company that's making drugs that can save lives is worth nothing, and a company that's mainly for sharing gossip is worth billions. Okay, so that's the world we live in, and so something has to be done about that. But the problem is, no matter what you do, whatever drug you make, eventually resistance will evolve, and this is just a consequence of natural selection. And the way that it can evolve is in many different ways. If you look at these antibiotics, they all work because they're a tight fit between the antibiotic and its pocket in whatever its target molecule is. For example, the ribosome. Now, you could destroy that tight fit in a variety of ways. The bacterium could make enzymes that would break down the antibiotic. So now the antibiotic is like two pieces. Each piece doesn't bind very tightly in the pocket because it's sort of too loose. It doesn't form all of the bonds needed for a tight fit. Or you could modify either the pocket or the antibiotic by adding extra chemical groups to it. So there are enzymes which do both of these. Or you could mutate the pocket so that when you have a mutation, then the shape of the pocket's different. And this again destroys that perfect shape, perfect match between the antibiotic and its pocket. And then there are also pumps in bacteria which will pump out antibiotics before they can harm it. The point is, whatever the mechanism, eventually resistance will evolve. Brand new drugs against HIV, eventually resistance evolved against all of them. So that's why they have to now take triple antibiotics to slow down evolution of resistance. So it can't just be making new drugs, okay? That's not the way to keep up with this thing. So we have to have a multi-pronged approach. So we have to have good surveillance. So when there are epidemics, for example, this is the job of the CDC and other agencies, you have to have very good surveillance so epidemics are identified and curtailed. You have to promote rational use of antimicrobials. I forget whether there were about 10 million prescriptions for the common cold in the United States per year, okay? When everybody knows that, antibiotics are not effective against the common cold. People go, or the flu, people go with the flu and ask for antibiotics. Antibiotics are widely used to fatten up cattle. I mean, this is a completely, terrible use of antibiotics, okay? Because that's a great way to spread resistance among, microbes which can then be transferred. Something is wrong here. Oh, sorry. So, and then you want to have good infection control. So in Britain, the incidence of MRSA deaths in hospitals declined by a huge factor when the National Health Service required everyone entering a ward to wash their hands, okay? Whether they were doctors, nurses, visitors, before you entered the ward, you had to sterilize your hands. And finally, there are a number of ways scientists can help. They can understand how bacteria cause disease, which is different in every case. And so you can have very specific targets. You can have better diagnostics, so you know which bug you have, so you target your antibiotics specifically for that. You can have try and develop vaccines. And the last part is what I talked about, which is trying to develop new target-based drugs and using the ribosome as an example. Now, I just want to close with what a fascinating machine the ribosome is. I showed you a very simple version. This is a slightly more sophisticated version. So you can see that the ribosome starts off with a small subunit trying to figure out where to start on the messenger RNA, which is this long, snaky molecule. That requires 3 different factors in the first initiator tRNA. And then the large subunit combines and then these factors go away. And that leaves the ribosome at the beginning of what's called the elongation cycle, which is where the protein chain keeps getting elongated by adding amino acids together to it. And that's done by bringing in tRNAs. That actually requires another protein to bring it in. And if it's the correct tRNA, then the tRNA is accepted. And then it goes in, and in this pocket is where the two amino acids are linked up. And then the whole assembly has to move. So the tRNAs move, and that's a two-step procedure. Well, it's a multi-step, but two major steps. First with respect to the large, and then with another factor helps to push it with respect to the small. And that brings the next codon in place, and then the next tRNA can come in and bind and so on. And then the first tRNA leaves. Now, this whole process takes place at the rate of 20 per second. If I showed you that, it would just be a blur, okay, because a movie is about 24 frames a second. So I've slowed it down by a factor of 5 or so. And you can see the growing protein chain comes out of the ribosome and then folds into the shape. Remember those 3 completely different proteins I showed you? Each one had its characteristic shape. And the shape is, the information for the shape is contained inside the chain itself. And then finally, when it gets to the end, a special protein comes in and cleaves off the newly made protein so it can go off and do its thing. And there are thousands of proteins being made in every cell, and each of them is made by this sort of complicated process. So I'd like to stop there. Thank you very much.
So the format that we typically follow is, please sit down, and there will be questions from the audience, from Ether Space, and perhaps I'll make up some questions. So let's start. Maybe are there? Yes, sir.
Is it now known like how the ribosome decides its priority? For example, like if a cell needs to make 5 proteins at a very fast rate at the same time, how does it decide its priority?
That's a major problem in molecular biology and has been for the last 30 or 40 years, which is gene regulation. That's what you're talking about, the regulation of gene expression, which is how does a cell know when to make a protein, how much to make of it, and how to stop making it. So you know those proteins I showed you, like hemoglobin, which carries oxygen in your blood. Well, you don't have hemoglobin in your brain, in your neuron cells. You know, you only have it in your blood. Or collagen is found in your skin, but it's not actually found, you know, inside the cell, for instance. Or rhodopsin is only found in your eyes. It's not found anywhere else. So how does that happen? Well, that's a multi-step procedure. The cell has sophisticated mechanisms for regulating transcription, which is making the copy of messenger RNA from the gene. And that's one of the steps where it's controlled. Another step where it's controlled is what's called translational initiation. So when the ribosome has to start translating, the efficiency of the way it begins translating can be controlled by the cell. And that can also lead to more or less of the protein being made. And finally, there are lots of steps which promote degradation, so degrading either the messenger RNA or degrading the protein, either more rapidly or less rapidly, is another essential ingredient, because you can make things at some rate, but you can also degrade them at different rates.
So can I go back to one of the points you made about resistance? Again, being from a physics perspective, what is it that you can learn or say it's dramatically different when you try to understand the flow of electrons or phonons, for example? Fermion versus bosons. Is there anything that you can translate from solid systems into understanding resistance?
No, I think resistance is best understood as a as a principle of natural selection. So you have genetic variation due to mutations and new properties being acquired all the time. But normally, there's no way these particular bacteria, so if you take a colony of bacteria, there are a huge number of variants in them. But normally they're silent because you're not selecting for any particular trait, okay? But it's only when you apply selection pressure, so only the bacteria that have a particular trait will survive under those conditions. Then you're selecting for those bacteria that have randomly acquired those mutations. Okay, so if they've acquired, for instance, enzymes that will modify compounds by adding acetyl groups or methyl groups, so that's a classic way of acquiring resistance. Or if they're bacteria that have modified the ribosome binding pocket, they've acquired a mutation. Normally, it doesn't matter. It doesn't do anything. The ribosome still works and so on. But suddenly, if you add antibiotics, you'll find that only those bacteria work, okay, and can live and survive and divide. And so now you have a colony of bacteria, all of which are resistant to this antibiotic. And then they multiply, and then they can pass on these resistance genes to other bacteria.
So let me reset. Perhaps we shouldn't hammer this question, but it's kind of piquing me. So in organic systems, for example, I can make a material which is very resistant, perfect insulator, right? And with time, silicon dioxide is a good example.
Yeah.
Right? It'll start to break down. Now, we know how to understand the breakdown mechanism, whether it's tunneling or whatever else. Can you use those kinds of ideas to go back and try to understand how resistance breaks down? Or is it already understood?
I think this is a slightly cultural problem, okay?
Let's explore that.
Physicists think in terms of design, okay? So this is not something that's designed. This is something that's selected for, okay? And so it's a completely, there is no physics equivalent. There are other cultural problems with physics as well. For instance, physicists don't know about controls, okay? Physicists have no idea.
Guys, wake up call.
Physicists, for instance, when they do an experiment, don't have this concept of controls. The reason biologists have to have controls is because biological systems are intrinsically messy and complicated. So when you observe a phenomenon, you want to make sure that it's due to what it is that you think it's due to. So you have to design all sorts of control experiments. If you do a physics experiment, you've defined the system so carefully that the need for controls, doesn't exist. So this is another, situation.
Yes.
Sorry? It's interesting, but almost every area of the ribosome that's functionally important has antibiotics that attack it. For instance, the site where the protein bonds are made, the peptide bonds are made, lots of antibiotics there. The tunnel through which the chain emerges, lots of antibiotics there. The place where the tRNA has to be recognized, lots of antibiotics there as well. So, virtually every important part of the ribosome seemed to have a natural antibiotic targeting it. And that's because there have been, a billion years or so for these bacteria to develop all these compounds. And, so in the process, they've sort of hit most of the possibilities. But companies are looking to see if there are pockets that they could attack for which there are no natural antibiotics. So it's not, inconceivable that you might be able to, this would go back to a more of a design philosophy. It's not inconceivable that you could think of a pocket, knowing how the ribosome works, and try and design a compound that would block that pocket.
Klaus.
Yeah. I have a question to some strategies to slow down the development of resistance against antibiotics. And I've read about that, one of the problems is to, what you also say, is we need to dramatically reduce the use of antibiotics and target them more. But what about some inflammations on which we don't know exactly if they're coming from they identify strep, and they immediately get an antibiotic. While I've also read that about 30 to 40% of all the kids always have strep in their throat, and it's not causing inflammation. So you get a dramatic number of false positives that way. So how could we reduce the cost of doing a much more targeted identification?
Yeah. So, for instance, E. coli, is a very common laboratory organism. Probably half of molecular biology studies have been done with this single organism, which was isolated from the sewers of London, I'm told. Anyway, but the point is that E. coli is normally, the lab strains we use are not harmful at all, okay? But there are virulent strains of E. coli, and that's, what causes these outbreaks, when you go to some hamburger place and people got sick and so on, So it's the same organism, and if you were to just do a general diagnostics test for E. coli, you wouldn't be able to distinguish between, some benign E. coli that's just, part of your gut versus some virulent strain. So I think we need better diagnostics. We need diagnostics that are very specific for pathogenic strains, and that's coming. I think that's going to get, lots of companies are investing in these very easy and fast chip-based assays, for, these kinds of infections.
Ma'am, you had a question there.
The problem is that we're going to constantly go after these molecules that fit into these little pockets. And the problem is that bacteria then have a mutation and then they find a new pocket, right?
Or they find a way to make it useless. Yeah.
So why not go to the source and use bacteriophages to then attack and mutate on purpose areas of their DNA?
Yeah. So I don't know how many of you have read Arrowsmith by Sinclair Lewis? Oh, that's a shame. This guy was one of the great American writers, okay? Anyway, Arrowsmith is about exactly that. This is about, the hero is a guy who goes to what really is a thinly disguised version of the Rockefeller Institute, which is now Rockefeller University, and works for a guy who tries to develop phage-based antibiotics. And the problem is, so far it hasn't worked, okay? Bacteria also can very quickly become resistant to phage. And then, there's a question, small molecules you can package nicely into a pill, and you can study how quickly their uptake is and how they're secreted, et cetera. If you have a bacteriophage, it's this large virus, effectively. It's a virus that attacks bacteria. It's not clear how you would control it and how you would introduce it into the bloodstream where the bacteria are and so on. So there's a problem of delivery, there's a problem of dose, and there's a problem of resistance and so on. So for whatever reason, and I'm not an expert in this area, it hasn't worked, although it was thought of, even in the 30s, okay? And I think people are constantly keep coming back to it. But so far, anyway, it hasn't quite panned out.
Yes, sir.
So before we had antibiotics, mammals actually used a mechanism to fight tuberculosis T-regs. So there's only about 4 to 6 degrees centigrade difference in temperature. And there are multiple issues which are involved here, cytokines and other things. But ribosome function has also been implicated. So could you say, do we know anything about the mechanism?
You mean by elevating temperature, the ribosome function of what? Of the bacterium or the human response?
So basically the thought is you're doing something to the machinery of the bacteria, basically, which has to live outside the body. So it's not thermally regulated at whatever the human temperature is. But the bacteria has to survive, in cold temperature. So it's optimized more for a wide range rather than a particular temperature. So there's some investigations which are looking at the difference of how ribosomes work under increased temperature for the bacterial machinery.
Yeah, I've spent most of my time looking at fundamental mechanisms, But that's different from rates, okay? So the rates the rates do vary quite significantly as you get away from the optimal temperature, which is typically 37 degrees, which is our body temperature. And E. coli, which is a human, symbiont normally, also grows at 37 degrees. So its optimum is also about 37 degrees. And as you get away from it, it is true that that the ribosome function may deteriorate. It may slow down and not be as efficient. It possibly may be prone to errors. I'm not sure of that. So it's possible that fevers are a mechanism to slow down the bacterium in relation, while you mount an attack. Okay, I mean, I wouldn't hazard a guess to what all of the evolutionary mechanisms are for why we get a fever, what the benefit is of having a fever. It could be that the fever is also just a reaction, and that fevers are also dangerous beyond a certain limit, but maybe they have a certain benefit up to that limit in trying to do that. I'm not really an expert in the area, so I can.
So can I go back to a couple of social questions? First, tell the audience, is there an algorithm to aspire and win the Nobel Prize, or is it seriously? I mean,
it's if there is an algorithm, it's going to lead to a life of unhappiness. Okay? I think just what happens is most of my life, nobody ever. I don't think the idea of a Nobel Prize ever. Well, the Nobel Prize entered my mind on two occasions. One when I was a teenager, okay, and thinking of going into science. Okay, and many teenagers who are going into science fantasize, hey, maybe I could get a Nobel Prize. But, that's adolescent fantasy, and when you start doing science, you stop thinking like that. Okay? And then what happens is that suddenly, your work has reached a level where you go to meetings or you go to give seminars or talks, and other people start saying, this could get a Nobel Prize, and so on. And it's actually a terrible thing, because then you start thinking, hey, maybe I could get one, and then, it's a recipe for disappointment, because, the Nobel Prize was founded in 1901, okay? And in 1901, imagine how big the science establishment was and how many scientists there were, and they were mostly all in Europe. There were very few scientists in the U.S. in 1901. And, all of them worked by themselves and, in isolated groups. They didn't communicate. And so 3 prizes, 3 people per discipline per year seemed okay, for major discoveries. But, nowadays, first of all, there are lots of discoveries. And secondly, it's becoming harder and harder to identify the real source of each discovery, because what happens is it's not like you're working 5 years in isolation and then suddenly you go to a conference, to solve a conference in physics or something, and announce this big, new idea or new discovery. You're publishing a paper, you're emailing friends, you've got a web page where all the data are, and you're going to meetings. People are going to far too many meetings now, and so your germ of an idea is very quickly disseminated, and other people are, using that to do their thing, and so it becomes very diffuse. Discovery becomes very diffuse. So I think people should probably just do what they like and find interesting. That's my.
Brian?
Is it known how biological function changes? This is kind of a physicsy question. If you go from hydrogen to deuterium?
Yes. So, for many higher organisms, deuterium is toxic. Okay, so for instance, if you were to have, I forget what the level is, but if you were to have more than a certain fraction of D2O in your blood, you would die. Okay? But on the other hand, you can adapt E. coli and even somewhat more complicated organisms, usually unicellular, to almost 99.9% D2O, essentially fully deuterated. Okay, and I think part of the problem is that many reactions involve proton transfer, and those reactions, the kinetics are affected if you have a proton or a deuteron.
So can we then follow up because Brian brought up the issue of deuteration, perhaps you could comment on why you chose electrons over neutrons or any other source of radiation to probe the structure?
Oh, that's very simple because crystallography, to solve large unknown molecular structures could not be done by any other technique than X-ray crystallography until 2 years ago, until a year and a half ago. And that's because, first of all, you need extremely strong fluxes, okay, and you need to be able to measure these reflections very accurately. And secondly, you need a way to solve the phase problem. I slightly cheated when I said you could recombine the rays, because what you're measuring is only the height of the waves, how high the trough or how low the cleft is. You're measuring what's called the amplitude of the wave. But you're not measuring where each wave is, where each crest is in relation to all the other crests, which is the so-called phase problem. And that required, very special methods. So in my case, I used a technique called anomalous scattering, where certain elements at particular wavelengths of X-rays would cause a difference between two normally symmetry-related reflections. Okay, so it's a highly technical reason why you could only do it by X-rays. You couldn't use electrons until recently because the detectors weren't good enough, and there were problems with correcting for movement, and the algorithms weren't good enough. But largely due to developments in my institute, spearheaded by Richard Henderson in my institute and a young colleague named George Sherris who did the algorithm part, we've now been able to solve by microscopy almost atomic resolution structures. So now there's a second technique, and which is for large structures, it's going to take over from crystallography.
Malcolm?
So doing protein crystallography, you have a lump of something which is static, and then you look at it and you say, well, this is the structure. But then for what you really want, and what you were showing in these presumably simulations at the end, is dynamics.
Yes.
Now, could you elaborate how you get to that, and then
how
reliable are the dynamics that you think you're watching?
Okay. So if you solve the structure of the static structure of the ribosome, even with the mRNA and tRNAs bound, and you want to make that movie, there's no molecular dynamics program in the world that would get you that movie, okay? But what we had to do, so what I've been doing for the last 15 years, so the first structures were solved in 2000, and here we are in 2014. So for the last 14 years, what we've been doing is figuring out biochemical ways to trap the ribosome at different points along the machine's workings, okay? So we have the ribosome in the act of delivering a tRNA, after it's delivered a tRNA. And then last year, we caught it halfway moved, okay? And then in 2009, we had it fully moved all the way. And this involves movement of the subunits, etc. So this is like getting snapshots of the movie, okay? Now, getting snapshots of the movie will give you a movie, but it won't tell you how the ribosome goes from state A to state B. But now you know what A and B look like, okay? Because you've trapped B and you've solved its structure, you've trapped A and you've solved its structure. But you still don't know how it got from A to B, okay? And that's where molecular dynamics can help. And the more finely tuned snapshots we have, the better chance molecular dynamics has to tell us how to get from A to B.
Take two last questions. Sir.
From the standpoint of you brought up the free electron lasers, and you can see single cells, single molecules. What fraction, in your experience or people you talk to, of the materials cannot be crystallized and studied by the ways which we all know and use, but would rather be extraordinarily important to be able to solve with a single shots at a
yes. So I think the big technique of the future in structure of large biological complexes is going to be electron microscopy. And there are several reasons for that. And one of them is that you don't need crystals. The second is that you need very small amounts of material. You need about 1,000 times or even less. So if you were to set up your crystallization trials, and then when your tube was empty, you just put a little bit of solution in your tube and just took whatever's left on the side of the tube, that'd be more than enough for your electron microscopy experiments. Okay, that shows you how little you need. And the third and very important thing is that it's now possible to computationally sort out a heterogeneous sample. So if you have a mixed population, let's say I had ribosomes, not all of them had reached a certain stage. Some were in one state, some were in another state. Okay, and we've actually done this. And we showed, so last year we published a paper in Science where we showed that the structure we were after was only 3% of the sample. Okay, only 3% of the molecules in the sample had the structure that we were looking for. And we were able to identify it by these sorting classification techniques. So I think that's one way to go. Free electron laser they're showing that it's useful when you have very small crystals, nanocrystals. But so far, they have not been able to solve an ab initio structure without crystals. And I'm not even sure they've solved an ab initio structure with crystals. Okay, so I think, I would be glad to have some of my tax money go for the free electron laser, but it's still at the stage where, it's being developed. It's more promise now, yet.
One last question. Yes, sir.
Thank you for this very nice talk. I just have a very general question. What I understand, bacteria develops its resistance to antibiotics through mutation.
From what?
Bacteria grows resistance.
Can you use the mic?
Yeah. So bacteria goes resistance against antibiotics through mutation.
Yes.
So I guess the same thing will happen to a human system as well. That also mutates. So I mean, in the process. So does that? Do you consider this as well when a new drug is developed or an effect of antibiotics?
You mean mutations in humans?
In the human system as well?
Yeah. Well, you have to realize the doubling time of a bacterium is about 20 minutes, and the doubling time of a human is typically, used to be 20 years. Now it's about 30 years. Okay, and so, it's not likely that if you take an antibiotic for a week, it's going to have any effect at all on so it's not going to really select for us. I don't think so.
So join me again, thanking Venki for a fantastic talk. Hold on, guys. Hold on. Don't leave. There's a formal presentation. Sit.
We'd like to offer our thanks for giving this Eugene P. Wigner Distinguished Lecture in 2014. This is sort of heavy, so we're happy to mail it to you. You don't have to carry it all the way back, but thank you very much.
Thank you very much.
And watch this space, because the series is not done.
Yes.
We have more great lectures coming, and I look forward to seeing you all there. Thank you.